Hello dear students. Till now we have already studied about two experiments. One was Griffith's experiment, other was Hershey and Chase experiment, which proved that DNA is the genetic material. But the question arises, what makes DNA a genetic material? Is only DNA a genetic material, or other molecules like proteins and carbohydrates, can't they act as genetic material? So, to get the answer, today we will study about the properties shown by the molecule for being a genetic material. So, you can see that I have written 4 properties. Any molecule, if it shows all these 4 properties, it can act as a genetic material. Okay. So, number 1 is the molecule should be stable enough, which means it should retain its property throughout the life of an organism. It should not break down and change in the lifespan of any organism. So that is stability. Second is ability to mutate. Mutation is a sudden random errors. What is the advantage of mutation? In fact, mutation gives a chance for evolution, chance for change, so that the organism can adapt with the changing environment. Third is it should express in Mendelian characters, which means a specific segment of a genetic material should be clear for a particular trait, expressing a particular trait. So, the third point says that any genetic material or any molecule can be considered as genetic material if it expresses in Mendelian characters, which means it controls specific traits in the body of the organism. Then fourth is generate a replica. Genetic material must pass on from one generation to next. If it cannot pass on from the one parent to the offspring, it cannot act as a genetic material. So, these 4 properties, if you compare, especially in 3 molecules, that is protein, DNA and RNA. Let's find out whether these 3 molecules show these properties or not. So, let's begin with the first property in detail. Stability, as I told you, the genetic material must remain stable throughout the lifespan of that organism. Not only that, it should have the ability to disintegrate and reintegrate. That is, when you heat a molecule, it might show denaturation, but when you cool it, it must show renaturation. This property is most suitably shown by DNA. Proteins fail in this property because when you heat a protein, it will denaturate. The bonds will break. But when you again cool it, it cannot renaturate. Whereas the DNA and RNA, they show this property. Okay. So first, in terms of the first property, DNA and RNA qualify to be a genetic material. Then second is ability to mutate. As I told you, mutation is a random change which might occur because of many reasons. Though most of the mutations are very harmful for the survival of that organism, but sometimes these mutations might prove to be a basis for evolution. If you compare mutation, RNA shows a very fast rate of mutation. There are two reasons for it. Number one, in RNA, the pentose sugar is ribose, which has got a free OH group. And this free OH group is highly reactive. Also, RNA has thymine as one of the nitrogen bases, which is also more reactive. Whereas in case of DNA, though it shows mutation, but the mutation is comparatively slow. There are again few reasons. Number one, the pentose sugar present in DNA is deoxyribose, where there is only H in place of OH, which is not as reactive as OH. Then the uracil, which is a highly reactive nitrogen base, has been changed to thymine. Okay, so which is more stable. Also, DNA is double helical, which shows complementary base pairing. As a result, it is always double helical and shows more stability. So, the mutation rate is comparatively less. Now, third is expressed in Mendelian character. So, when you talk about a gene is a segment of DNA which are controlling the expression of a character. So, both DNA as well as RNA, they qualify for this trait. That is, they express in Mendelian characters. They control specific characters. Okay. And last one is generate a replica. Both DNA and RNA has the ability to show replication. And that replication process is very well defined in DNA, which is called as semi-conservative mode of replication. Whereas even RNA, it has the ability to show the replication. Okay, so both DNA and RNA, they can generate their replica. So, even many scientists believe that the first genetic material on earth was actually not DNA. It was in fact RNA. But this RNA, being very, very reactive, comparatively very less stable, it showed very fast mutations. And these very fast mutations were not suitable for the longevity of the organism. So, it is believed that the RNA converted into double helical structure and became the DNA. Still, there are many reactions which are controlled by RNA. Only DNA is replicating, but most of the reactions are still revolving around RNA, like transcription, where DNA is first converting into RNA for protein synthesis, and then that RNA is again diversifying into mRNA, messenger RNA, tRNA, transfer RNA, rRNA, ribosomal RNA, which we will study later on. And then it follows translation, where whatever information is coming from DNA, it is converting into the form of protein. That is called flow of information. Also, it is believed that the first enzyme on earth was not a protein. It was an RNA in the form of ribozyme. So, scientists believe that the first genetic material in fact was RNA, but for the stability and for controlled mutations or for to reduce the mutations, RNA converted into DNA. So, I hope you must have understood that what makes any molecule a genetic material. So if you compare these characters, it is very evident that both DNA and RNA qualify for being a genetic material. Protein is not a genetic material, but if you compare the stability, it is the DNA which is preferred as genetic material. Only few organisms have RNA as genetic material. That is in some bacteriophages, not in bacteriophages, mainly in plant viruses, few animal viruses and viroids. Otherwise, all other organisms have DNA as a genetic material. Okay? Thank you very much.